Cell death is an integral part of our body's systems and functions, yet death is almost always associated with being bad for us. Programmed cell death is one of the most important fail-safe systems that our body has. This process is called apoptosis, and it is essential to a healthy organism. But the question still remains, what is the purpose of this mass cell suicide if one of the main drives of an organism is to stay alive?
Multicellular organisms have highly complex and organized ways of regulating all of its functioning units. Having control over the rate of cell division is just as important as controlling the rate of cell death. In adult tissues, cell death exactly balances out cell division. If not, consequences such as tumors can form.
A cell that undergoes apoptosis dies neatly, without damaging its neighbors. The cell shrinks and condenses. The cytoskeleton collapses, the nuclear envelope disassembles, and the DNA breaks up the nuclear DNA into fragments. After the cell dies, a signal is sent to neighboring cells to digest the remaining fragments.
Let's take a look at a classic example of a signal that controls apoptosis. P53 is a tumor suppressor that encodes a gene regulatory protein that is activated by damage to DNA and is involved in blocking further progressions through the cell cycle. MDM2 is a protein that typically combines with P53 as a P53-MDM2 complex. When a cell's DNA is damaged, P53 will separate from MDM2. After dissociation, P53 protein either undergoes apoptosis or triggers a DNA repair process. It is hypothesized that apoptosis will happen when the DNA repair process fails after cell cycle restart. It is a switch determining the cell's fate.
Another main protein involved in cell death is the death-associated protein kinase, also known as DAPK. It is an intracellular protein that mediates cell death by its serine and threonine kinase activity. This activity transmits apoptotic cell death signals in various cells, including neurons. DAPK functions as a tumor suppressor, but its expression is lost in many cancers due to mutated receptor methylation. DAPK acts in a cascading manner like many other different cell pathways do. Its major target is the tumor suppressor P53. DAPK-induced apoptotic cell death requires P53, and a positive feedback system results from this relationship. DAPK will induce P19ARF, thereby inactivating MDM2 to then stabilize and activate the P53 tumor suppressor.
In a DAPK mutant, the activation of DAPK by ERK was eliminated, causing it to lack a death domain. This example highlights a mechanism through which the death domain may be necessary for full DAPK activity in cells. P53 is not the only mediator of DAPK-induced apoptosis. ERK1 and ERK2 are also mediators. Interestingly, a reciprocal regulation between DAPK and ERK has been revealed. The death domain kinase interactor, ERK, phosphorylates SIR735 to activate DAPK, causing an increase in DAPK catalytic activity. Conversely, DAPK blocks ERK nuclear translocation. Since the function of ERK in promoting cell survival is mainly associated with its nuclear localization, retention of ERK in cytoplasm would block ERK's survival signal. Thus, the ERK-DAPK complex constitutes a positive feedback loop to promote apoptosis.
Cell death is a necessary event within these organisms to target and destroy individual cells that will present a threat to the integrity of an organism as a whole, as demonstrated in this video. Cancerous tumors forms when cells are constantly being proliferated or the apoptosis process is slowed down or halted due to mutation. In the field of oncology, scientists look to understand the mutations in these proteins that control cell proliferations and apoptosis. If we are able to generate a specific protein phosphorylation pathway to activate the apoptotic pathway, we can slow down the proliferation of the cancer cell.
Restoring endogenous P53 function holds a lot of promise. Research has been done to show this restoration can lead to regression of certain cancer cells without damaging other cells in the process. The ways in which tumor regression occurs depends chiefly on tumor type. With restoration of endogenous P53 function, lymphomas exhibit apoptosis and cell growth is lowered to normal levels. Loss of P53 creates genomic instability that most often results in the aneuploidy phenotype. Thus, pharmacological reactivation of P53 presents itself as a viable cancer treatment option. Gaining knowledge from researching proteins such as P53 can help reach the long-term goal of finding a cure, because everyone deserves a lifetime.